Um, Today we're very happy to have here Troy Neptune that did the PhD in Western uh, Case Reserve University, and then moved to Cleveland Institute of Art for doing his first postdoctoral stay. And during this time, he's visiting us for a Fulbright postdoctoral grant with Katarina Ruthratz with me. He's an amphibian ecologist with a strong interest on the effect of light pollution, photoperiod, and temperature on different life history traits of amphibians. And actually, Troy now got a very nice postdoctoral position at Oberlin College that is going to be started very soon, probably in July. So thank you, Troy, and good luck with the seminar.
Thank you, Pablo. So let's get the obvious thing out of the way, which is, yes, I'm wearing the same shirt. No, that was not planned. And yeah, no, I did not take the picture today. This was a month ago. But you know, hey, some things work out. Yeah, so we're going to be talking a lot about how amphibians respond to light, photoperiod as like this ecological cue, and then disruptions to that cue via light pollution. So we'll talk about how photoperiod and temperature affects tree frogs before and after metamorphosis. We'll then move on to effects of photoperiod on thermal physiology as these tree frogs prepare for winter. And then we'll finish by talking about some of the work that we're working on here at the EBD on effects of light pollution on spadefoot toads. So before we dive in, if there's any questions throughout the talk, please let me know. Please interrupt me. That is totally fine. If you need me to repeat anything, please also just shout that out. So let's start with photoperiod and temperature effects on tree frogs. So we're going to be talking a lot about photoperiod today. Photoperiod, I'll say it probably 100 times during this talk. It's really important that we all understand what that is. So photoperiod is the duration of light in the day or the day length. All of you are probably familiar that in the summer we have longer photoperiods or longer days. In the winter we have shorter days or shorter photoperiods. And just to really hammer this home, today in Sevilla the photoperiod is 14 hours and 9 minutes. On this date, which is May 14th, on May 14th, every year at this location, the photoperiod is always 14 hours and 9 minutes. That does not change from year to year. It's actually one of the few things that's not changing due to climate change. So we have this really amazing consistent cue that lots of organisms respond to. And so all kinds of different taxa use photoperiod to time different behaviors throughout the year. So for example, you know, birds migrate at certain times of year. They're breeding at different times based on photoperiod. Even within the same day, you know, we have nocturnal versus diurnal organisms. And so photoperiod is super important from just day to night and then also across the year. But one of the problems is climate change. So this was a video, but now I have a before and after. So that's the before. This is 1881. This is the year 2023. So we all know in here that climate change is happening. The world is heating up. And so there's lots of problems that arise from that heating up of the Earth. But one of those interesting problems is that many organisms are advancing their phenologies at different rates with rising temperatures. Phenology being this timing of important events in an organism's life. And so, for example, we have the spring ephemeral. This spring ephemeral is emerging earlier and earlier over time because global warming. So as temperatures are rising, the spring ephemeral is coming out earlier and earlier every year. And so one problem with this is that organisms that shift these different phenologies, whether that's emergence, breeding, migration, whatever, due to temperature, will then experience novel photoperiod patterns. And for example, what I mean by that is if we have this cute set of implectant tree frogs up here, if they typically breed in mid-June, perhaps over time with shifting their breeding phenology due to temperature, we see that maybe eventually they shift their breeding, you know, to June, to May, to April. And so if they were to breed in April, that represents a very different perception of photoperiod. And so those larvae that would typically be developing under a June photoperiod, if, let's say, the parents are then shifting their phenologies over time, which gray tree frogs are, if they've shifted way earlier to maybe in April or May, that ends up being this pretty significant change in photoperiod. So for example, you have from mid-June to April, this change of about 2.5 hours of light. And so these days are shorter, right? Because this is earlier in the year. Summer, we have our longest photoperiods with the summer solstice. And so we had this big change of 2.5 hours of light. So it presents this interesting question of what happens because of that. So this isn't the only example. Some tree frogs and some different species may not be adjusting their breeding phenology at all. And so if they do not change their breeding phenology, then you have this increase in temperature because of, again, global warming. And so if they're not changing their timing of their breeding, those larvae are exposed to higher temperatures. We all know this for our different organisms that we study. And so in this case, if they don't shift their breeding phenology, these larvae are developing under the same photoperiod conditions. So now this sets up this kind of interesting experiment, which we'll dive into, of how do these tree frogs respond to photoperiod, how do they respond to temperature, and then how do they respond to any synergistic effects of photoperiod and temperature. So why photoperiod? What do we know about life history in photoperiod? Well, for many different organisms, lots of damselflies and dragonflies and other ectotherms, we have faster metamorphosis and reduced size under late season photoperiods. This is a trade-off. So under different photoperiods, for example, under a late season photoperiod, this damselfly is going to metamorphose faster, and because they're metamorphosing faster, they're going to come out at a reduced size, right? That's our classic time and size trade-off. Now, this is likely an adaptive response to a cue indicating the end of the growing season. So if the damselfly can detect via photoperiod that it's coming to the end of the season, well, it's time to breed, it's time to, whatever other species, it's time to migrate. And so it's an adaptive response for them to be able to detect photoperiod and then respond appropriately. What do we know about how amphibians, though? We're here to talk about frogs. I'm a frog person. What do we know about how amphibians respond to photoperiod? Well, like I mentioned, many amphibians are breeding earlier in the year, and we have some very limited evidence, including the first chapter of my dissertation, for larvae developing faster under shorter photoperiods, so a reduced amount of light compared to another treatment. The problem, though, is photoperiods are short at both ends of the year. So this is, if this is across time, right? So we have April over here all the way down to September. We have the day length over here. And so short photoperiods, you have on this date in May and then this date in August, these are actually identical photoperiods. They're both 14 hours of light and 10 hours of darkness. And so that's problematic because these are the same amount of light. So how do organisms know which part of the season that they're in, right? Are they in spring or into summer, or are they in the late summer going into fall? Well, the difference here is that these photoperiods in May are increasing because we're heading towards the summer, right? So days are getting longer, the photoperiod is increasing, versus in August, the days are getting shorter because we're approaching winter and the winter solstice. And so the question becomes, can organisms detect these marginal changes in photoperiod each day, and is that an important cue for different organisms? So we're also interested in temperature, right? Because we're concerned about climate change, and we're concerned about this question of temperature versus photoperiod. We know that lots of good data, this amazing figure from this great paper that I've cited multiple times, and I have it in all my presentations. This is Katarina Ruthstadt's part of her dissertation, right? So I use this figure all the time. I've known about Katarina for quite a long time, as well as Pablo. So we know that from lots of different studies, we have different taxa, different temperature treatments and things Amphibians develop faster under warmer temperatures during the larval period. And so as temperature increases down here, we have a decreased age at metamorphosis, meaning that they're developing faster. So we have a pretty good understanding of amphibians and other ectotherms are developing faster under warmer temperatures. But the question becomes, after exiting the pond, after they metamorphose and leave the pond, how does the larval thermal environment affect juveniles post-metamorphosis? And so this is what we would consider a carryover effect. How does conditions in one life stage carry over to affect a later life stage? So this brings me to my research questions for one study that I'll walk through with you. Do photoperiod and temperature have comparable effects on amphibians before and after metamorphosis? And then two, do photoperiod and temperature have any synergistic effects that we need to understand in the context of shifting phenologies and global warming? And I'll do this, lots of my research has been on the eastern gray tree frog, a very cute frog, probably one of my favorites, although I'm a little biased since my whole dissertation's on the eastern gray tree frog. So the eastern gray tree frog is pretty widely distributed across the eastern United States. Has anyone in the audience seen an eastern gray tree frog before? Okay, there's a few people, yeah. I'm used to like a lot more people seeing that in the US. They're like, oh yeah, they're in my pool in my backyard. So eastern gray tree frogs are pretty widely distributed. Something that I want to point out here is that we're doing this study up here in Ohio, but as you can see, they're widely distributed across latitude, and so changes in latitude mean changes in photoperiod, and so this is an interesting species to investigate effects of photoperiod because they are so widely distributed across latitude. So I've shown you this a few different times. This is an example of the photoperiod treatments that we used in this particular experiment. So we used photoperiods that started on April 1st, June 21st, and August 15th. So, across the breeding season of this species. To show you a different way of looking at that, so we started our experiment. We have days since the start of the experiment on the x-axis, starting from 0 and beyond. We have our different photoperiods here in different colors. In purple, we have our early season photoperiod that started on April 1st. In the yellow, we have our late season photoperiod that started on August 15th. And then in the teal, we have this June 21st photoperiod, our average photoperiod that's pretty long and then slightly decreasing over time. We had our frogs in these different treatments, so early, average, and late season photoperiods. We crossed that by temperature, so we had a warm temperature treatment and a cool temperature treatment. We did this under field conditions, actually, so I didn't have any electrical power, so I actually fastened together these little artificial greenhouses to help warm our mesocosms in the field. And so we have all of our amphibian larvae in these different tanks across the field, and we use these greenhouses to help warm up the temperature of the water, and I had lots of help from two amazing undergrads. And then we also manipulated photoperiod. We can get more into that later if you're really curious, but I manually manipulated photoperiod using black plastic to basically cover the tanks, and we used a clear plastic as a control. So we had our larvae in our photoperiod by temperature treatments in the field during the larval period. As they metamorphosed, we moved them into the lab, and we actually put them under the same photoperiod and temperature conditions in the lab to isolate effects of the larval period to look at carryover effects of temperature and photoperiod on post-metamorphosis. We put them through a 10-day growth period just to look at some short-term effects. And we had a smaller subsample that we kept in the lab for a little bit longer, almost 2 months, to look at slightly longer-term effects. So let's start with our data at metamorphosis. So we're going to be looking at a lot of these figures. I'll help orient you a little bit. So we have photoperiod on the x-axis here. We have our early season, average, and late. So April, June, and August. On the y-axis, we have age at metamorphosis. So this is how long these animals are taking to metamorphose. And so essentially what we're seeing here is that frogs from the shorter photoperiods, so this is early and late, and the warm temperature treatment are metamorphosing faster. So you can see in the red we have our warm temperature, so we see this nice response that's pretty expected. The frogs are metamorphosing faster under the warmer temperature. But then we have this effect of photoperiod where if they are in that early or late season photoperiod, they're also developing faster compared to the average photoperiod. So that was age at metamorphosis. The next figure looks pretty similar, but now we're looking at mass at metamorphosis. So how big these frogs are when they finally metamorphose. And so we see that frogs from the shorter photoperiods, the early and late season, are smaller at metamorphosis. And then the frogs from the warm treatment are actually larger at metamorphosis. So this is kind of the opposite of what we might expect there. And so for the photoperiod, they have this response of developing faster and they're coming out smaller. So that classic trade-off that we've talked about earlier. That's a little different from our temperature treatment where they developed faster and they achieved larger sizes when they were warmer. So there's some various reasons for that, and we can talk about that if you'd like later on. But we have this interesting independent effects of photoperiod and temperature. There weren't any interactions here to discuss. So we also saw with this combination of faster metamorphosis and a larger size at metamorphosis, that means if you do a little bit of math, we have increased growth rates. So the frogs from the warm temperature treatment grew faster, but then we did not see any effects of photoperiod on growth rate. Okay, so that was our data at metamorphosis. Does anybody have any questions at this point? Yes.
Which was the temperature you used in your treatment, cool and warm? Which target was the temperature?
Ooh, it was a, the change in temperature was 1.3 degrees. So not a big change, but enough. Yeah. And that's an average because they were under field conditions. Yeah, exactly. Okay. So let's move on to our data on 10 days after metamorphosis. So we looked at the short-term growth rate. And we were surprised to find that frogs that came from warm larval conditions during the larval stage actually grew about 4.5 times slower post-metamorphosis over 10 days. So if you recall, they were growing faster up until metamorphosis, but then when we looked at 10 days post-metamorphosis, they actually grew a lot slower compared to the cool temperature treatment, which is pretty interesting. So that was 10-day growth rate. Let's take a look at our longer-term growth rate. So we had our short term, now we are looking at our long term. Here we have no impact of temperature at this point, but under this longer time span, we actually see this kind of emerging response where juveniles from the shorter photoperiods, the early and late season photoperiods, actually grew about 2 times slower compared to those that came from a long photoperiod. And so we have this interesting effects that there were no effect of temperature on this longer term growth rate, but then we had this kind of latent or emergent effect of photoperiod on that longer term growth rate that was pretty significant, and 2 times difference is quite large. And that's the result to show you that on 56-day growth, we saw this effect, but then in previous time points, we did not see this effect of photoperiod. So what this means is that at the end of the experiment in the lab, we had juveniles from the longer photoperiod were larger after 56 days. So you can see that over here we have our photoperiod treatments, final mass on the y-axis, and we have our frogs that came from the average season being pretty big compared to the other 2 treatments at the end of the experiment. So this is I collected a lot more data, not to overwhelm you. What I just want to show you is these triangles. So essentially what we're seeing is that if frogs are in that early or late season photoperiod, they're essentially responding in the same way across all kinds of different metrics, morphology, timing of metamorphosis, all these different things. And so this is actually suggesting that perhaps this is the cue that they're responding to, that maybe, we talked about how these photoperiods are short, both in the early and late season. So perhaps these frogs are responding to the total amount of light rather than this directional change in light over time. We also did some cool thermal physiology stuff. This is actually some of the impetus for why I will show you the data coming in the future slides. So we had these frogs in the lab, and I thought, well, it would be really cool to understand some of their behavior. And so we put them in these thermal chambers. You can see that we have some ice packs down here. We have some heating mats down here. And so essentially this chamber is a thermal gradient representing about 10 degrees Celsius to 30 degrees Celsius on the other end. And so what we did was we put the frogs in the center of these chambers. We allowed them to select temperatures over the course of a few hours. And then we're recording their body temperature with infrared thermometers. And so we're getting an idea. These frogs are allowed to freely move within these chambers and select temperatures that they would select. And so what we see here are that frogs from the late season photoperiod selected cooler temperatures compared to those from the April or early season photoperiod. So this is their photoperiod at metamorphosis. We have our late season down here. They're choosing about a 3.3 degrees Celsius lower temperature compared to those in the early season photoperiod. So pretty interesting. They're under the same temperature and photoperiod conditions in the lab at this point, but when they're put into a thermal gradient, they're selecting about 3 degrees or more difference in temperature. So that was pretty cool. So this brings us to some of the conclusions from this part of the talk, which is that frogs from the shorter early and late season photoperiods metamorphose faster and are smaller, while frogs from the warm treatment metamorphose faster and are larger. We see evidence for frogs responding to the total amount of light rather than this incremental change in light over time. And we saw that early and late season animals responded in the same way. And then this kind of gives us this idea that under future climate warming, when we're thinking about that short-term growth rate after metamorphosis, frogs that grow slower post-metamorphosis may be at a disadvantage. And so right after metamorphosis is a very sensitive time period, and so not growing as fast during that period could indicate something about their longevity. Okay. And if you're interested in reading more about this work, you can check out that QR code. Okay. So that was this kind of first part of the talk where we're talking about photoperiod and temperature effects. There weren't any significant interactions between photoperiod and temperature. We actually generally saw that there were these just kind of additive effects of both variables, so not having this synergy between the two variables. And actually what we found was that under our experimental conditions, we argue that photoperiod was actually perhaps more impactful across a suite of traits versus temperature. So we saw these effects on behavior, morphology, timing of metamorphosis, all due to photoperiod, less so due to temperature. So it's this interesting thing going on with photoperiod, of which we've had very limited evidence about until now. So now we're going to talk about freeze tolerance in frogs. This is my favorite chapter from my dissertation, so we're going to talk about that next. So like I mentioned before, the world's heating up. We all know this. But with that warming, we're getting these warmer or shorter winters. And so this is the US. And so this is over time, starting from 1890 to 2020 and more. And so essentially what you're seeing here is that the spring frost, the last spring frost is happening earlier and earlier. Here we have the first fall frost is happening later and later over time due to climate change. And so we're getting these warmer or shorter winters in the continental United States. Also everywhere in the world. So this is interesting because temperature and photoperiod are becoming mismatched. And so photoperiod is this cue that's tightly linked to temperature. If the days are getting shorter, organisms can perceive that the winter is coming, and so they can respond appropriately. Photoperiod is this really consistent cue from year to year like we've talked about, but that's becoming less and less true because the climate is warming. And so I was curious about how tree frogs use photoperiod as they prepare to overwinter in terms of their thermal physiology. And then I wanted to ask, are there any kinds of costs associated with this overwintering preparation? And so we used a similar setup to the previous experiment where we had these 3 different photoperiods. We had our early season here in purple starting on April 1st and then ending on June 29th. We had our photoperiod, our average season, starting on July 2nd, ending on September 29th, and then our late season photoperiod starting on August 15th down to November 12th. And so what I want you to pay attention to are the photoperiods on the right side of the figure. These are the ones that are going to be pretty important for the thermal physiology that we're going to be talking about. And so we have frogs that are experiencing a photoperiod kind of in the peak of the summertime, really long days. We have the beginning of fall here in September, so days are starting to get shorter. And then we have frogs that are in the lab perceiving the photoperiod to be in November, which is at that point they should be starting to settle in for the winter in Ohio. And so we'll first talk about some critical thermal physiology. So I know some of you are familiar with CT max and CT min. I'm sure some of you have run frogs under these different kinds of experiments before. Critical thermal maximum, or CT max, is the highest temperature before an organism loses motor function. And so you heat up animals until they stop moving. Hopefully try not to kill them. We don't want that. And then you can get their CT max. The inverse is also true for CT min. You want to cool animals down. So critical thermal minimum is the lowest temperature before an organism, or the point at which they lose motor function. And so you can cool down animals until they stop moving or stop behaving, and then that is their critical thermal minimum. So we have, I wasn't just doing this experiment just for fun or just on some sort of hunch. I did have a little bit of data from a previous study that suggested that there was some lower critical thermal physiology going on here with gray tree frogs, a sister species actually of gray tree frogs, under field collected adults. So under field collected adults, they saw that there was this change in CT min towards the end of the season. So that was interesting to me. I wanted to do an experiment to investigate this further. And so, at this point in the season, August 28th is where we're at. Could it be, are frogs responding to a change in temperature if the days are getting colder? Is that what cues them to start putting on antifreeze reserves and starting to overwinter, or is it a change in photoperiod? And so what I did for the CT max and CT min trials, I had a hot water bath, which was great. We basically put these tree frogs in the hot water bath. We gradually increased the temperature of that water bath over time. We kept heating them up until they stopped moving. And then when they stopped moving, I flipped them over to make sure that they couldn't right themselves. I then confirmed that that's the temperature at which they could not move. And then I flipped them back, and then I brought them back down to room temperature. And I am proud to say that I have not killed any frogs during that experiment. And so we were heating them up. They were fine. Everything was good. And then we did the same with critical thermal minimum. I did not have the luxury of a cooling water bath, so I did the ice method, if anyone is familiar with that. You add a certain amount of ice, you cool down the water at a pretty consistent temperature, and then you see, same thing. We flip the frog over, make sure that the frog can't right itself, and then you record the temperature, right? And so the ice method, worked out fairly well. And I got their CT min. So this is my little nod to my Spanish colleagues. Thank you so much. So we saw that under the late season photoperiod, these frogs actually exhibited a greater cold tolerance. So they were in the lab. They were under the same thermal conditions in the lab. And we had them under their photoperiod treatments in the lab. And we actually saw that there was this effect of photoperiod on thermal tolerance, which is pretty cool. It's one of the few examples across ectotherms broadly. There's an example in mealworm larvae, you know? So certainly the first amphibian example of photoperiod affecting thermal physiology, one of the only vertebrate examples as well, if not the only one at this point. So we saw that under the late season photoperiod, they selected, or they had these cooler critical thermal minimums. So essentially, if they were under the late season photoperiod, so that, what did I say, November photoperiod, they could actually tolerate colder temperatures. Why is that important? Well, if it's becoming the winter because it's late in the season, being able to tolerate a lower temperature would be a good thing, right? Because then you can select micro-refugia and you can survive the winter and things like that. We did not see an effect of photoperiod on CT max, but we also had the opportunity to describe CT max for these juveniles, which was novel in its own right, but we did not see an effect of photoperiod on CT max. Okay. Any questions on critical thermal physiology while we're here? And I can take a sip of water. Nope. Okay. So now we're going to get to my favorite part, which is freeze tolerance. So I don't know, being here in Sevilla, I don't know how familiar you all are with freezing tolerance, but I love talking about freeze tolerance. It's actually this pretty widespread trait across different taxa, beetles, fish, salamanders, turtles, intertidal snails, and then also, of course, frogs and other amphibians. And so all these different kinds of organisms can exhibit freeze tolerance. And so we're going to be talking about frogs, of course. So you might be concerned about these two frogs. They're covered in ice crystals. Oh no, they're fine. They're going to thaw out in the following year and they will survive because they have this amazing ability to mobilize cryoprotectants or antifreeze, such as glucose and glycerol, to withstand freezing conditions. And so these are some freeze-tolerant species, gray tree frog, wood frog over here. And again, I did not do this on a whim. We had a little bit of information to suggest that, okay, cryoprotectants or antifreeze can accumulate seasonally and can be freeze responsive. And so in this paper, I think this was wood frogs, maybe it was gray tree frogs, but they had this decrease in glycogen, which is the cryoprotectant that we'll talk about, or the precursor to the cryoprotectant that we'll talk about. They had a decrease here, and so there was some indication that perhaps cryoprotectants are accumulating across the season, but really no information on why, until now. So cryoprotectants are really great. They prevent intracellular ice from forming and prevent cells from exploding. So that's how they function, and that's the extent of the physiology. I'll tell you about that. So what we did, I worked with this really amazing collaborator at the Cleveland Metro Park Zoo. We collected these frogs. We determined their glycogen concentrations on liver tissue. And so this is an overwhelming amount, so we'll start with that piece first. So essentially what we're seeing here, we have our different photoperiods here. And what we'll see is that we have glycogen concentration on the y-axis here, which is our amount of cryoprotectant or antifreeze. Glycogen is then converted into glycerol, which is then circulated throughout the body as an antifreeze. And so we were looking at glycogen concentration in liver tissue. We saw under the late season photoperiod, so again, these are frogs that are in the lab perceiving the photoperiod to be November 12th or early November. We see that they have these really concentrated tissues for glycogen. And so there's a lot of glycogen in these tissues. When you take a look at their livers, this is final mass of the frog on the x-axis, liver mass on the y. And basically, when you take a look at this, those in the late season, again, over here in the tealish color, at a given body size, these frogs have huge livers. So you would dissect these frogs, and they're all liver, these giant livers. So when I did that, I was like, oh, that's pretty exciting, because I feel like something's going on. And then when we take a look at doing a little bit of math here, multiplying liver and glycogen concentration, you essentially get this huge concentration or huge amount of antifreeze in these bodies of these tree frogs, all based on photoperiod. So under this late season November photoperiod, they have about 14 times the antifreeze compared to the other treatments. Pretty cool. But this did not come at no cost. These tree frogs from the late season photoperiod grew about 28% slower post-metamorphosis, which was interesting. So it suggested that perhaps they were using photoperiod to slow down, maybe not eat as much. What they are eating, they're packing on as antifreeze, getting ready for the winter. So this brings us to the conclusions for this part. Gray tree frogs under the late season photoperiod had these greater cryoprotectant reserves. They had a lower critical thermal minimum, and they also reduced their growth over about 1.5 months. The kind of subtle thing that I haven't really mentioned is that this was in the context of warm conditions in the lab. These frogs were kept at, Fahrenheit to Celsius calculations, let me think, like 20 degrees Celsius in the lab. So these frogs were in pretty warm conditions in the lab. Certainly not under realistic conditions for the winter. The winter is very cold in Ohio. We didn't want to freeze the frogs in the lab. So they were kept under these warm conditions and still responded in this way. So truly they were having this response to photoperiod regardless of the fact that the lab was pretty warm. And so like I mentioned, these photoperiod photoperiod becoming mismatched with temperature, and I propose that this might become an ecological trap for these frogs, where if they're responding to photoperiod instead of temperature, then they might prepare for the winter too early because photoperiod and temperature are becoming misaligned. And so you can read more about this at Journal of Animal Ecology. Photoperiod has these lasting impacts on amphibian growth, morphology, development, and behavior. And we saw these strong responses to photoperiod. And so these responses may become maladaptive in a warmer future because photoperiod and temperature are becoming increasingly mismatched. And so any organism, not just frogs, the ones that you study as well, if they track photoperiod instead of temperature, because of climate change and global warming, they may fall into an ecological trap where their responses are no longer adaptive. So something to consider for all the species that you all study as well. So that was my antifreeze part of the talk. Now we'll talk about spadefoot toads, because I know there's some toad people in here. And this is the work that I've been doing at the EBD. It will continue for a little while longer after I'm gone. And so this is pretty cool. Now we're going to talk about disruption of photoperiod due to light pollution. And so light pollution is getting worse every year, about by 10% each year globally. The US has lots of light pollution, so does the Iberian Peninsula, so we're all at fault for this. This is important because I don't have to explain to all of you that there are lots of species that are threatened by extinction. I'd like to highlight amphibians. About 41% are threatened by extinction. It's the most threatened vertebrate taxa. So that's why I care about amphibians in particular. Light pollution, we'll be talking about ALAN, artificial light at night. This is the use of artificial light during the night in human society. We use it all the time. You may recognize the center of Sabia here. Very pretty during the winter time, but there's a lot of light pollution. You can hardly tell that it's supposed to be nighttime because of all of the lights. This is a problem because ALAN can cause mismatches between seasons and organisms' circadian rhythms. And so what happens is when there's so much artificial light, that actually increases the perception of photoperiod. And so organisms that encounter artificial light have a disruption to their photoperiod because it's light. They get confused. They think it's daytime. And that can lead to all kinds of different impacts on migration, metabolic rate, and things, and different aspects of their physiology and behavior. So ALAN is disrupting this perception of photoperiod. So you can have birds that are singing earlier than they're supposed to. Bats are coming out at different times than they're supposed to. And all kinds of things can go wrong. To have a few more concrete examples, bats are abandoning their roosts or delaying their emergence due to artificial light. Birds are famous for having disrupted migration due to artificial light at night. And then insects are also famous. They get, their flight patterns are all messed up because of artificial light. And so to hammer this home, over 50% of key biodiversity and protected areas around the globe are exposed to light pollution. So it's something that we need to be thinking about and doing more with. But we really don't know a lot about how light pollution impacts amphibians. So we have a little bit of information about some stress responses and things, but there's really, little research on the impacts of ecologically relevant levels of light pollution. These are some great studies, but there are a few of those in there that expose amphibians to really high levels of light pollution, and so that may or may not be biologically realistic. In the experiment that I'll walk you through, we're using ecologically validated levels of light pollution that we know are occurring in wetlands around the world right now. And so we're doing this work with the spadefoot toad. We were interested in how does Allen photoperiod and temperature influence resource allocation and metabolism before and after metamorphosis. And then we are also curious, this is the part that's going to go on while I'm gone, how long and to what extent does exposure to low levels of Allen during the larval stage persist after metamorphosis, so that carryover effect of artificial light at night. So the toads are here. A lot of you know about these toads. They breed in habitats like this. Just to walk you through the design of the experiment. So we have a pretty complicated experiment in the lab, which has been amazing. We have, and a lot of work, but we have these different levels of light pollution. So we have toads that are not exposed to Allen. We have a low level of Allen, which is about 5 lux. And then we have a high level of Allen at 30 lux, which both are ecologically relevant levels of light pollution. We cross that with photoperiod. So we have a long photoperiod, and then we have a short photoperiod. Because that wasn't enough, we also crossed it with temperature. And so we have all these treatment combinations. We have toads developing at 24 degrees Celsius and toads at 27 degrees Celsius. So we have this big, giant experiment in the lab, which is pretty amazing. And so just to show you what this kind of looks like visually, so this is our no Allen treatment. No light is hitting these toads at night. During the nighttime, we have this 5 lux, and then we have 30 lux over here. So these are perhaps brighter than you would expect, but these are ecologically relevant levels, like I've said. And so we have 2 different photoperiods. We have our long and short. We have a photoperiod that had started on December 21st. We have a photoperiod that started on March 21st. You can see here that this is similar to the other figures that I've shown you, but we have days since the start of the experiment. Here on the x-axis, we have the day length on the y. And so we have our toads under these 2 different photoperiods. We have our long and short starting in December. Right now they're currently in about mid-March. And then the other treatment started on March 21st, and they're in about mid-June, so the longest days of the year for these toads. So this represents different ends of the breeding season, different breeding phenologies that they are naturally exposed to. The data that I'll show you is collected about right here, so mid-larval stage. We also had these 2 different temperatures, like I've said. If you want to go see the chambers, go walk over to the chamber room. There's chambers there. If you want to look at the experiment, talk to Katerina or Pablo first. You can't go in there without their permission, please and thank you. But it's a pretty awesome experiment. Very lucky that I get to do that here. So we have our larvae in these treatments. We have them in our Allen by photoperiod by temperature treatments. After they metamorphose, we're planning to keep them under their temperature treatments in photoperiod conditions, but we're removing that Allen exposure to isolate the carryover effects of artificial light at night on some different post-metamorphic traits. So we'll talk about what we have going on during the larval period. We're looking at lots of different traits. So what I'll first show you is that tadpoles are actually developing faster under the short photoperiod in higher temperature, but there's no effect of Allen exposure on development rate at this point of the experiment. So again, these larvae are about mid-larval stage. So we're not seeing, we have our Allen treatments down here. We have no Allen, low level of Allen, and then high Allen. And then on the y-axis, we have development rate. And so, and then we have our photoperiods here in the orange circles are the long photoperiod. The blue triangles are the short photoperiod. And so we're not picking up statistically an effect of Allen at this point. Stay tuned. There might be some effects on metamorphosis. That's data to come. And so, but we do see an effect of photoperiod where if they are under the short photoperiod, they're developing slightly faster. And then, of course, at the higher temperature, we're seeing faster development rates. So all the toad people that are in the room, there's effects of photoperiod on your toes developing, which is pretty cool. When we take a look at growth rate, we see that tadpoles are growing slower at the higher temperature, but we're not seeing an effect of Allen or photoperiod treatments on growth rate at this stage of the experiment. So we also dissected these tadpoles to understand liver size, fat deposition, and gut size. So what we found here on the kind of resource allocation fat deposition side of things, we see that tadpoles have larger livers under the short photoperiod and lower temperature, but then there's not an effect of Allen on liver size. And so we're seeing that under the short photoperiod, they have these relatively larger livers, which is pretty interesting, but no effect of Allen. And I forgot to mention that on the left side, we have our 24 degrees C animals, and then on the right side, we have our 27 degrees Celsius animals. But what we now see is that the panels have changed. We now have fat body mass on the y-axis. We see that tadpoles have more fat bodies under the short photoperiod and under the no Allen treatment, but there's no effect of temperature. So what we see here is if they're under the shorter photoperiod, they have more fat in their bodies versus the long photoperiod. And then we also see this effect of Allen treatment where if they're exposed to low or high Allen conditions, they have less fat bodies in their bodies, which is pretty interesting. They also have heavier guts under this no Allen exposure and also the cooler temperature. No effect of photoperiod here. So not only do they have more fat bodies under the no Allen treatment, but they also have heavier guts, meaning they might be, these guts could be longer, they could just be eating more, things of that nature. We also took a look at metabolic rate, and this was a lot of work and lots of hands involved in this process. The metabolic rate was pretty cool, but we did not find anything on the effects of Allen or photoperiod on metabolic rate for these animals. Data is only available for those at 27 degrees Celsius, so not really seeing an effect here, at least, on metabolic rate. So this brings me to my summary as I'm running a little tight on time. We saw these clear effects of photoperiod on development rates and resource allocation. And so tadpoles under the short photoperiod, they have higher development rates, and I think that will continue to be true for metamorphosis because they're metamorphosing right now and have some ideas there. And then we have greater fat storage in the livers and fat bodies due to photoperiod. And so bigger livers, more fat bodies, also developing faster under the short photoperiod compared to the long photoperiod. And then exposure to Allen, we saw that there were some effects on fat deposition and gut size. So tadpoles under our low and high Allen treatments had decreased fat bodies and decreased gut size, which may be related to fill. Something I want to point out is that if there's less fat in these tadpoles, this is the important point, that there's less available energy for metamorphosis, there's less available energy for early survival during post-metamorphosis. It's a very critical time for frogs and amphibians and these toads. So having more fat is a good thing. And so if they're having less fat due to Allen, that could indicate some sort of problem going on. Like I said, there are lots more data to come. We have the toads are metamorphosing right now, so we're working on finishing up the experiment here in maybe another month or so or more. We'll be analyzing some corticosterone data during both the larval stage and post-metamorphosis to understand impacts of Allen on stress levels. And we'll also be examining some responses during post-metamorphosis to understand how long these effects persist. We'll be looking at metabolic rates, seeing if there's any latent effects of photoperiod and Allen. And so wouldn't be the first time that there's latent effects that don't emerge until later. So it's important that we check these out during post-metamorphosis. Okay, so that brings me to, that's all that I have for the light pollution part of things. There's lots of people to thank. Obviously, lots of the earlier stuff that I showed you was from when I was at Case Western Reserve University. Lots of people helped out there. Special shout out to Fulbright and Fulbright España for allowing me to be here. I wouldn't be here without the Fulbright program. It's an international program that's sponsored by Spain and the US that allows me to be here, which is pretty amazing. And of course, thank you to the EBD for hosting me. Obviously could not have done this work without this institution. And then the Toad team, so lots of great people involved with these projects. You'll see them, they're all here in the room. So thank you all for hosting me and helping out with the project. It's been pretty amazing. And at this point, I think we have some time for questions. Thank you.
Thank you very much for giving this nice Troy.
Thank you.
Do we have any questions for Troy?
Very simple. Just in your experiment that you're explaining that you have running right now, at the very beginning, like at time 0, what are they? Are they eggs or are they tadpoles? Are they all the same age, or how does it start?
Good question. So excellent question. So the question was about when did we collect them and what stage were they in? They were in very early larval stage. So we aimed to get eggs, could not find them, and so we dip netted and gathered larvae from the field from a big area with lots of different parents, so genetic diversity and all that. But they were early stage larvae appropriate for the start of an experiment. Yeah. Thank you for asking that. I didn't say that. So.
Do we have more questions? Thank you very much. It was a lovely presentation, very clear. Although you did a lot of complicated, experiments, so it was very well explained, I think. So my question is, with all your experiments, do you have an idea of how this may affect their population fitness or viability? Because also you talk about carryover effects. So for instance, if they are growing faster or slower on a certain photoperiod, would this mean that they then reproduce faster to compensate for it, or would this mean extinction or exacerbated extinction under global change in general?
Yeah, thank you. So great question. Long-term impacts, right? How does it scale up to species persistence? So effects of variables like temperature, photoperiod, artificial light at night, how that affects them, at metamorphosis and shortly after can really indicate how they're going to survive and persist in the terrestrial environment. And so we have, research to back up that frogs that are bigger at metamorphosis, they get bigger sizes at reproductive age, they survive better. And so being bigger at metamorphosis is important. So that's one metric. Getting there sooner or faster can also be important for survival and reaching sexual maturity faster. And then when we consider, post-metamorphic growth rate, metamorphosis is a really stressful time for these animals. And so surviving it and being set up for success for the terrestrial environment is really important. So for example, relating it to the fat deposition, if they have less fat, that means that they have less available energy. And so, perhaps these toads need to start eating faster, which might be challenging depending on the environment that they're in. And so having more resources as they enter the terrestrial environment right after metamorphosis could be really important. And so all these things we consider in the context of how that may affect them over the long term, like sexual maturity and reproductive size. And then of course, those things scale up to affect population persistence. And so, we have limited data on the effects of light pollution, but certainly some longer term studies will help. Studies like this will help give us an idea of how real populations may respond to these variables.
Thank you.
Hey Trey, thanks for the great seminar. I may have got lost in all of the results, but I had the impression that the responses of Hyla and Pelobates were contrasting in terms of the response to the photoperiod. So in one case, growth rate was enhanced, and the other way, in Pelobates' case, it was reduced. Is that correct?
So for photoperiod, we did not have an effect on larval growth rate. And so if we take a look at toads, yeah, I like this comparative question because I haven't really thought about it too much yet. So with growth rate, so we saw that there wasn't an effect of photoperiod, for example, on larval growth rate. That's consistent with gray tree frogs for the larval period.
Okay, well, but whether it's significant or not, it's like long photoperiods there seem to be associated with reduced growth, whether it's significant or not. And if I remember correctly, for Hyla, it was the other way around. Was it correct? So do you think it's like species-specific responses, or for instance, do you think in the mesocosms other things could have changed other than the frog response itself? Meaning, I don't know, because it's mesocosm out there. So are there like more phytoplankton or other stuff also responding to photoperiod?
Right. Yeah. And so first point is that under the post-metamorphic growth rate, we did see increased growth rate under the long photoperiod. And so I think that will be, it's different here, different life stages, but I think that will be an interesting point of comparison. Other question about the mesocosm setup, we did look at algae, phytoplankton, and periphyton, and the results are not very intuitive. They were weird. They did not explain any changes in growth rate. So they're a little complicated to interpret, and you would think that there would be more phytoplankton and periphyton under the long photoperiod because more light may suggest more algal growth. That was not the case. It was actually a different photoperiod treatment had more algal growth. But then, yeah, there are no effects of photoperiod on larval growth rate in the Hyla. So, yeah, but comparing between the lab and field experiments is interesting because we have to think about these other things in the field like algal growth, which we did think about, but it doesn't really explain any changes in growth rate that may have occurred.
Okay. Thank you. Thanks. So you brought up a very interesting issue, which is the mismatch of environmental cues and the response of organisms to that particular thing. And indeed, it's not warming, which it's normal that we are concerned about that, but it's the contradiction of all these signals. But I'm wondering, where are the boundaries of this mismatch in your study system? So are you considering experiments, to test, how far can organisms cope with that mismatch? So I say your Allen experiments, pointing to that direction, right? But what's your thought about that?
Yeah, so the one piece that I bring up there is this cryoprotectant part, because they are having this response in the lab at this relatively warm thermal environment. And so if we a priori expected the temperature in the lab to override the effect of the photoperiod cue, we would not see an effect on antifreeze accumulation here, but we do. And so we think that this photoperiod cue is pretty strong because they're having this response regardless of this warm lab environment. And so that's our one indication of this kind of, that question that you're bringing up. And so I would love to do a study where we did different, overwintering scenarios where we're manipulating temperature, we're manipulating photoperiod, and do a really involved study. It would be a lot of work, and I need the money to do it. But I would be happy to do it. But yeah, it's an interesting question. Yeah, the only thing I can show you is that they had these responses of photoperiod regardless of the fact that the lab environment was warm. Maybe they would have had even stronger responses if the lab was cold. I don't know. I'm not saying here that temperature isn't important, It could very well be, but what I am showing is that photoperiod is definitely important for antifreeze accumulation. Hopefully that helps answer. I know it's not a satisfying answer, but
We'll talk later.
okay.
Yeah, thank you. Thank you for your talk and very comprehensive results and so on. But yeah, regarding this same picture, what's about other antifreezer like glucose?
What about glucose?
What's about glucose concentration? Because glucose
Yeah.
So you have evidence of experimental evidence of increasing in glucose concentration after any event.
So we looked at, for gray tree frogs, it's actually glycerol is the primary cryoprotectant. We looked at blood glycerol levels. There weren't effects of photoperiod on blood glycerol. It was really low, and so we suspect that they weren't actually mobilizing their cryoprotectants yet because they weren't, there's evidence that, at least in this species, that they really need to experience that cold, that really, 0 degrees or below for them to actually initiate that change from glycogen to circulating glycerol in the body. And so, but we did look, and there wasn't an effect, but the levels were really low. So we suspect that they hadn't actually experienced an environment in which they would start ramping up glycerol production or glycerol circulation in their bodies. But it was a thing we wanted to make sure we checked because I didn't want to miss it with the glycogen and then it actually be circulating as glycerol. So we checked both. Definitely effects on glycogen concentration and levels, but not on glycerol.
Another brief question is, you are considering a single species that is within a community of tadpoles or amphibians in the ponds. What's about early breeder? Because Hyla versicolor is a medium late breeder, but what's about with the other species like wood frog, so that are more early breeder? Which effect you expect for the photoperiod, and what's about the possible mismatch in the competition level, interspecific competition level at this scenario?
Yeah, there's lots of open questions with photoperiod. With the comparison with wood frogs are interesting because they're explosive breeders, and so they have for everyone who doesn't know, they concentrate their breeding in a very short amount of time versus the gray tree frog, which has this kind of elongate, long breeding time. Same thing with toads, seems to be this longer breeding season. And so I would expect that these breeding seasons might relate to responses to photoperiod, because if you're breeding at different times of the year, months apart, then there might be some important adaptive responses to photoperiod. And in terms of antifreeze and things like that, I would suspect that it makes sense, right? That if you can detect that winter is approaching due to photoperiod, that you can ramp up, get ready for winter, accumulate cryoprotectant. So I would expect wood frogs to also have this response. No one's looked at it yet, but I would suspect that any, maybe any would be strong. I don't want to, you never say all in biology, but other species that have antifreeze may use photoperiod as a way to accumulate cryoprotectants to get ready for winter. Yeah.
Thank you for a very nice talk. I was just wondering, for the toad experiment here, how dark was the pond from where you collected those individuals? Do you think they would be adapted to artificial light already?
That's an interesting question. They were collected not by myself, by Katarina. Katarina, are there lights around that area? No. Okay, great. So no light pollution, but that is that's something we need to include in the manuscript for sure. Yeah, so no previous exposure to light pollution for these animals, and certainly during the larval, before they were collected, weren't exposed to light pollution, which is an important point. Thank you for bringing that up.
And also, so your treatment, how do they compare to moon phases, for example? So compared to a full moon, was the light stronger in your higher exposure, or would it still be less than a full moon?
Yeah, that's a good question as well. So moonlight, at a full moon, at most, it's 0.4 lux. Okay. So pretty, effectively pretty close to zero. For our treatments, we had 5 lux and 30 lux. So 5 lux, still quite a bit brighter than moonlight, but really not that dark when we consider, the street lamps that we have and all these things. So 5 lux is a really pretty low level. 30 is a bit higher, still both ecologically relevant, but much higher than moon phase or full moon lighting.
Yep. Cool.
And then the new moon is almost completely dark. Yep. I mean, it's very, very dim.
Yep. Yeah. Cool. Thank you.
Thank you.
Do we have any more questions for Troy? If this is not the case, Troy is around only for today. You are leaving tomorrow to the US. So if there are any questions left, make sure you meet Troy today. I think we are around in the climate chambers, but then unfortunately, time is running out. Yeah. Thank you again.
Send me an email.
Also. Yeah. Thank you very much for this fantastic seminar. And yeah.
Thank you all.
See you next week.